All right, so the next thing is DNA sequencing. So DNA sequencing is a modification of natural DNA replication. Dr. Sanger, who actually discovered this technique back in 1970s, used the kind of a mechanism of natural DNA replication and modified it to carry out DNA sequencing. Sanger is one of the scientists who actually won Nobel Prize twice. First time he won Nobel Prize was for his discovery of protein sequencing, and the second time was DNA sequencing. Actually, DNA sequencing is such a revolutionizing type of a technique that it changed the whole field of biology. And the human genome project that was completed in 2003 was based on DNA sequencing. And Sanger was the pioneer of that technique. Brilliant scientist. So DNA sequencing is to get the entire sequence of the genome. Big projects have been already completed, and in the center of this technique is chemistry. So Sanger was a chemist. He modified the nucleotides from being deoxynucleotides to become dideoxy. So this is natural nucleotide for DNA, but what he did was he did dideoxy, meaning there were removal of 2 oxygen. It's actually pretty neat. Let me show you where that is. This is dideoxy nucleotide. So in dideoxynucleotides, in natural, let me go back. In natural deoxynucleotide, there is, so this is carbon number 1, number 2, number 3 called primes, 4. So what happens is that it has back a little bit. So in natural pentose sugar or deoxyribose sugar, this one has hydroxyl group. This one has only hydrogen here. That's why this is called deoxy, meaning that one of the oxygen is missing here. Oxygen that should be here is not there. In case of RNA, RNA has the same pentose sugar as OH here and OH here. But in case of this is ribose for RNA, and this is deoxyribose for DNA. So in this case here, one oxygen is not there anymore. That's why it's called deoxy. So what he did, when Sanger did, he also removed the second oxygen from here. So when both of those hydroxyl groups are gone and are replaced by hydrogen there, what happens is that these dideoxys are not reactive. This group at carbon number 3 is used to make phosphodiester bonds that link one nucleotide with the next one. So when hydroxyl group is not there and is replaced by hydrogen, then this growth does not take place anymore. So in Sanger's DNA sequencing technique, so he will carry out this chemical reaction in which he will do basically 4 separate tube reactions in which, and all the 4 tubes, so he will have 1 tube here, 1 tube here, 1 tube here, another tube here. He will have all the components that are needed for DNA for DNA replication. That would mean, template DNA, would mean all the nucleotides, enzymes, and then one component that will be additional in those or unique in each of those tubes will be a dideoxy. Other than nucleotide, he will add only one type of dideoxynucleotide in it. For example, in this tube, he will have dideoxythymine, and everything else will be normal. In the other tube, he will have everything the same, but he will have one additional thing that will be dideoxy-C. And in the third one, he will have dideoxy-A. And in the fourth one, he will have dideoxy-G nucleotide in it also. So incorporation, so if you remember the DNA replication is based on a template, which is this one here, a sequence of DNA that is copied by filling the second nucleotides here. The second fragment or second strand of DNA is added based on the complementarity which is present on this strand on the top. Unfortunately, we are not really seeing the top template DNA here. So in one tube that has dideoxy-G, so when this process will take place, where the extension of that length of the template DNA takes place, wherever there is a G added, in some cases, the G will be a dideoxy-G. So meaning that it will not further replicate once the dideoxy is added. For example, we see that is being done here, being done here, and being done here. So because there are G's that can be added here in 3 places. So meaning that it will produce 3 different fragments of different sizes. Same thing will be done with A, because wherever there is A being incorporated, the chances are that some of them will have dideoxy added here, or here, or here, or here, and you will see all of them together. Same thing in case of C, same thing in case of T. So, and then he would pull them together in a single tube after he would do those separate reactions. Then he would pull them together and run them on gel electrophoresis. Now, the same thing that he did is being done, which is shown here in this image here, done with the help of fluorescence. So, for example, DDG is labeled with the fluorescence tag, which is blue in color. And then DDA is tagged with the orange fluorescence. C is tagged with green, and T is tagged with red ones. Now they become very sequence specific. And now you can actually pull them together, even though they have been done separately. Then you can pull them together and run them on gel electrophoresis inside the DNA sequencers, which are attached to the computer and all. Then, fully automated systems, they detect the signal that comes from the gel electrophoresis, and then they kind of photomultipliers will capture the signal that comes with the constant incidence of a laser beam, and converts that into a sequence in the computer. This event of gel electrophoresis taking place here is recorded, captured by the kind of internal mechanisms of the DNA sequencers machines, and convert that into a sequence that is recorded in the computer. So it's a fully automated system now. That's how most of the human genome was completed. Human genome project was mostly completed by this type of a fully automated kind of a fluorescent sequencing. This was the second generation of sequencing that we used. It was not really easy thing to do. Actually, I was at Cold Spring Harbor when this technique was started to be used in DNA sequencing. Our lab at Cold Spring Harbor was into sequencing. So that's were my first introduction about DNA sequencing. We did lots of sequencing of many different types of organisms, including humans and Arabidopsis and yeast and some other things as well. So we used to run the gels in the morning, or start the gels at night, and those gels will be done in the morning. Start the gels in the morning, they'll be done in the afternoon. So that was my life for what couple of years as a postdoc there. Fortunately, that technique got changed later on with more really fully automated DNA sequencing that kind of got the job done, which is the next generation sequencing. So next generation sequencing is much more robust and kind of completely hands-off type of techniques that produces a huge amount of data. So these are the kind of advantages. The disadvantage of the newer next generation sequencing is expensive. So not everybody can afford it. The hope was that the newer techniques after the first, second generation of DNA sequencing will be cheaper and could be used in the clinical settings, in the doctor's office setting, but we are not there yet.